Welcome to my talk on Trichoderma, a potential arsenal for management of soil-borne plant pathogen. In my talk, I will cover basics on Trichoderma, steps followed working with Trichoderma, long-term preservation technique of fungal biocontrol agent including Trichoderma species, compatibility with other microbes and agrochemicals, development of formulation, pilot field trial, demonstration and training, advantages of using Trichoderma based formulation, commercialization, and path ahead. Let me start with the basics of Trichoderma. As you know, Trichoderma is a free-living filamentous fungi which is also considered as an opportunistic and virulent symbiont that are used as biopesticide and are highly interactive in root, soils, and foliar environment. They compete for seed exudate, nutrients, and species. They are also used as a biofertilizer or fertility promoter. A specific strain of the genus Trichoderma colonize and penetrate root tissues as symbiont and lead to induced systemic resistance and help in stimulation of plant growth. Mycoparasitic mechanism together with effective defense mechanism induced in plant are considered as the key factor of the bioagent. They inhibit or degrade pectinases and other enzymes that are essential for plant pathogenic fungi for infection. During the last decade, species of Trichoderma have emerged as most powerful bioprotectant for management of wide variety of plant diseases. This is more true in the context of the fact that there is considerable public pressure and pressure from environmental scientists to reduce emphasis on chemical-based protectant and use protectants of biological origin. Trichoderma has long been considered as one of the promising agents for the management due to its broad spectrum action against fungi, bacteria, and even nematode, and occupied the top position among the bioprotectant developed for plant disease and pest management. Trichoderma strains exerts strong aggressiveness against phytopathogens either indirectly by competing for nutrient and space, modifying environmental conditions, or promoting plant growth and plant defense mechanisms and antibiosis, or directly by mechanisms such as mycoparasitism. Trichoderma added directly to rhizosphere or as seed treatment protect plant against numerous classes of pathogens. This reveals induction of resistance mechanism similar to hypersensitive response, systemic acquired resistance, and induced systemic resistance in plant. We started our work on exploitation of microbial resources, including Trichoderma species, with the following steps. We surveyed the prevailing agroecological condition of northeastern region of India and collected samples, isolated, purified, and characterized the biocontrol agents. The promising strain was selected through in vitro studies like dual culture test, parasitism test, enzyme releasability test. Then compatibility study was done with agrochemicals and other microbes. Rhizosphere competence was studied, followed by root colonization, and phytic behavior was also studied. Then we proceeded for activation of the biochemical defense responses. Then we tried the performance in the greenhouse as well as small scale field trial. Immediately after that, we tried for selection of the substrate for its mass multiplication and self-life study. Quality control measurement was also taken care of for propagal concentration, virulence, viability, contaminations, safety to non-target organisms. Then the technology was demonstrated and further refined based on the feedback received. We tried to popularize by different approaches and finally we commercialized the product. As I said, we have isolated characterized Trichoderma species from soil sample, plant sample, and the aerospora from farmers' field of the prevailing agroclimatic zones of northeastern region. A total of 70 isolates of Trichoderma were isolated, and the species were identified as Hergianum, Asperellum, Pseudoconynge, and Viridi. For preservation of fungal biocontrol agent, we faced problem as it need repeated subculturing that needs continuous involvement of research scholar, scientist, and then culture lose its viability and virulence after 3 to 4 subcultures. Moreover, lyophilization causes breakdown of the fungal spores. Ultimately, at field level, the bioformulation product shows erratic result. So we have developed one long-term preservation technique which we named as a Surakshit. And the cost involvement to preserve one culture in Surakshit is 2.25 INR. And in this way, we are preserving our culture and we applied for patent for the technology. We studied the efficacy against the following pathogens: Rhizoctonia solani to Melodogani incognita. Through a series of in vitro studies, which we conducted, and it ameliorated to identify the potential biocontrol strain of Trichoderma species. A study on mode of action of Trichoderma species showed that competition, mycoparasitism, lysis, antibiosis are the main mechanism of Trichoderma species for suppressing the pathogen. Our study also showed that seed treatment with Trichoderma hergianum at the rate 5 ml per liter can enhance the plant growth parameter with reduction of pathogen infection. But it was not known what happened when the trichoderma goes to the soil. It will definitely face the complex nature of the rhizosphere and face the presence of commonly used agrochemicals. So compatibility study of the trichoderma species were done with the above component. And during our study, we have found that out of 14 different fungal and bacterial biocontrol agents, except Pseudomonas and Bacillus, all other were compatible with Trichoderma species. And this helped us to develop one idea of consortial formulation with multiple actions. We tested compatibility with 4 fertilizer at 4 doses 0.1 to 2% and found compatible up to 2% except calcium ammonium nitrate that showed compatible up to 0.1% only. Compatibility with different stickers, adjuvant, osmoticants, spreader, UV protectant, preservatives were also done and found compatible with certain doses. During the compatibility test with the common agrochemicals against 10 insecticide, 5 fungicide, and 2 herbicide at lethal and sublethal doses, we found compatible reaction with 3 insecticide and a fungicide and as well as a herbicide at some doses. The good information is that this Trichoderma species has no harmful effect on beneficial insect like honeybee, predator like praying mantis, then earthworm and parasitoid like Trichogramma species. During the series of study, it was also found that the mighty fungus has also the ability to enhance the major secondary metabolite of plant like endochitinase, proteolytic enzymes, etc. It has the ability to enhance the plant growth parameter, ability of siderophore production, releases plant growth regulating substances, help in conditioning the soil and improve beneficial microorganisms in the soil, improve the uptake of micronutrient to the plant, especially copper, zinc, iron, sodium, etc. They also help in solubilization of the phosphorus in soil and make available to the plant. Moreover, application of trichoderma as an amendment with compost gives a better result than the traditional fertilizer. Then we tried to develop two protocols for bioformulation. One was solid, another was liquid, and supplemented with some sticker, carrier, adjuvant, and UV protectant. And during field study, we have found liquid formulation and was better than the solid formulation. These are some of the photograph of liquid formulation which having the shelf life of 16 months. We train the farmers, self-help group, farmers club, community interest group on technical aspect of bioformulation, method of application, preparation of spray solution. Distributed bioformulation to the farmers of Assam, Meghalaya, and other nearby states. We demonstrated the technology on method of application of biopesticide as a seed treating agent, soil application, as well as foliar spray. And in almost all the cases, we encourage more and more women farmers. Farmers use in different crop like paddy, citrus, tea, vegetable at different locations, and they found very good result due to the application of the bioformulation based on trichoderma. These are also some view of performance in the field condition of the farmers field. And here I want to mention that we are working in tea gardens and in one tea garden they could able to increase their area up to 450 hectare, and now it becomes a regular practice for using trichoderma based bioformulation for cultivation of the tea. This is another field where we have demonstrated the technology and successfully, and farmers convinced with the performance of trichoderma based bioformulation. We also tried the performance in the hilly terrain of Meghalaya, and even we also tried in the protected cultivations. Advantage of the product of the over the existing products are we prepared with indigenous strain, so it has high adaptability in our environment. It is safe to the environment. There is no question of residual problem. This formulation acts as a plant growth promoter. This formulation have a shelf life of 16 months. It has high demand in the tea as well as vegetable sector, and we have not recorded any contamination in our formulation. We tried to popularize through publication in the newspapers and talk in TV programs. Extension bulletins were also prepared in vernacular as well as English languages and distributed among the farming community. We conducted 10 days short course where scientists from National Institute, especially State Agriculture University, Central Agriculture University, and ICR Institute, they participated and they were trained on preparation of the bioformulation. Until to date, we have trained 1,400 farmers of 2 states, 80 extension functionaries, 120 scientists and faculties of different states of India, 60 tea garden managers, and distributed 1,000 liters of bioformulations. And it is my pleasure to inform you that as of date, these bioformulation are using in 8 different states of northeastern region of India. And we used to get the feedback from the end users, and they have reported that they got early bud break in tea at least 7 to 10 days. Unproductive tea bushes were seen with healthy flushes, higher antioxidant property in tea, then higher shelf life of vegetables were recorded with high value in the market. They reported enhanced plant growth and early flowering, and they could able to minimize the traditional fertilizer based on NPK and synthetic pesticides. The technology generated for plant disease management with bioformulation of trichoderma has been included in the organic package of the states. And the success story were covered by Indian Phytopath News and national newspaper like the Telegraph. The 4 product protocol, including trichoderma-based formulation, is procured by Green Biotech Eco Solution Private Limited. They have established 82 outlets in different states, and now they actually appointed unemployed youth, including agri graduate, and they have the target to sell their product in the nearby country like Myanmar, Bhutan, and Bangladesh. Another company, they are using the strains isolated by our group for preparation of the commercial formulation as per MOU. In the coming days, we will try to produce some microencapsulated products so that we can give the farming community a handy and ready to use product. And in the near future, we have to do more studies to determine environmental effect on the fat of bioagent like trichoderma, integration of the biopesticide like trichoderma with botanical pesticide has lot of potential in pest management, enhancement of the potentiality of biocontrol agent through protoplast fusion, and this type of microbe can be utilized for synthesis of green nanoparticle like silver, copper, and gold. So, last but not the least, I would like to thank my farming community for their support, my funding agency, my student friend, and my family members for their continuous support. And my special thanks goes to Dr. S. Santra, Director UCF MISA Center, who invited me for this talk. Thank you. Thank you all.